Hello everyone, and welcome to the first video of the Burlington Central Brain Bee Club YouTube channel. My name is Josephine, and in this video I am going to be breaking down the first chapter of the Brain Facts textbook. Brain Facts is a resource commonly used for students preparing for Brain Bee, which is a neuroscience competition. And the first chapter is just covering basic neuroscience content. So I'm going to do my best to break that down for you in this video. So with that being said, let's jump into our chapter 1 mini lesson. So the first kind of theme covered in that textbook is brain divisions. These are the main divisions. So at the very front of the brain, we have the frontal lobe. Behind the frontal lobe, we have the parietal lobe. We also have the temporal lobe in green. And at the very back, we have the occipital lobe. A couple things to note, this brown structure underneath the occipital and temporal lobe is called the cerebellum. And this stalk-like structure is called the brainstem. We will be talking about the brainstem shortly, so something to keep in mind. But the parietal lobe deals with processing sensory information like that of vision, touch, and taste. The occipital lobe processes visual information, and specifically when it comes to shape recognition, facial recognition, and object recognition. It's unique because the eyes are located at the very front of the brain underneath the frontal lobe, yet the occipital lobe, the part of the brain that processes visual information, is all the way at the back. So that might be something you can use to help you remember its function because it's a bit unique. The frontal lobe deals with decision making, problem solving, memory, emotion, personality, and speech. It's the last part of the brain to fully develop, typically at the age of 25. And down here, the temporal lobe deals with audio interpretation and language comprehension, specifically when it comes to the Wernicke's area, which is in the temporal lobe, language comprehension and being able to understand speech is where that would happen. Moving forward, like I mentioned, we have the brainstem. So sitting just on top of the brainstem is the thalamus. It's technically not part of the brainstem, but it's typically included in a lot of diagrams. Underneath the thalamus, we have the midbrain. We also have the pons, and we have the medulla oblongata. So the midbrain, pons, and medulla oblongata are all part of the brainstem. The thalamus is the sensory relay station. This is where sensory information stops to be relayed to a specific area in the brain to be processed. So it's kind of like a pit stop. For example, visual information. It's going to come in from the optic nerve, from the eyes. It's going to stop at the thalamus, and the thalamus will redirect that information to the primary visual cortex in the occipital lobe. Okay, the midbrain has two sections. We have the superior colliculus and the inferior colliculus. The superior one deals with visual reflexes. So that's your blinking reflex, your gaze, your pupillary dilation. We also have the inferior colliculus, which deals with auditory processing. The pons is a relay station between the brain and the cerebellum. The cerebellum is that brown structure that I talked about underneath the occipital and temporal lobe, and it deals with movement, coordination, and balance. So damage to the pons can potentially result in impaired movement, even if that's coming from alcohol abuse. That can also affect the pons. The pons also deals with facial expressions, breathing, and sleep. And the medulla oblongata deals with your autonomic functions. Those are breathing, heart rate, blood pressure. These are things that are vital to your survival. So damage to the medulla oblongata typically leads to a quick death. Moving forward, there are just a couple more structures I want to highlight in the brain. This is looking at these specific structures internally from another angle. So again, we have the thalamus here. We also have the hypothalamus, which has a slightly different function from the thalamus. We have the frontal lobe. We have the olfactory bulb, the amygdala, and the hippocampus. The hypothalamus has everything to do with homeostasis, keeping balance and rhythm within your body. So that has to do with your circadian rhythm, your sleep, arousal, body temperature, aggression, thirst, hunger, and so on. The olfactory bulb is the initial smell processor. So this is kind of where smell is initially processed, and then it goes through the olfactory nerve into the olfactory cortex. This is the only sensory pathway that does not go through the thalamus, which makes sense because it would just be easier to go straight to the olfactory cortex because it's so close to the nose. The amygdala is the emotional center. This is where your fight or flight response is happening. This is where all of those kind of emotionally heavy things are going on in the brain. And the hippocampus is the memory center. This is where your short-term memory is converted to your long-term memory and it's being consolidated. So these are some key structures to be mindful of as you move forward in your preparation for Brain Bee. Another common theme that was talked about in Chapter 1 is brain waves and electroencephalograms. Careful not to mix that up with electrocardiograms, which are for the heart. So brain waves, there are different kinds, but this is essentially looking at the level of activity of the brain. And that changes depending on if you're awake or asleep. If you are awake, you can either have what we call alpha brain waves, which are 8 to 13 hertz. Hertz are cycles per second, so it's just the frequency of brain activity. And then you can either have beta, which is 14 to 30 hertz. If you are asleep, you can have either theta brain waves, which are 4 to 7 hertz, or delta brain waves, which are less than 3.5 hertz. And then at the top on that diagram, we have gamma brain waves. So alpha brain waves are typically when you are restfully awake. So your eyes are closed, but you're not sleeping, and you're also not really thinking about anything. So you're just kind of, relaxed, hanging in there. Beta is when you are focused or you are processing sensory information. Theta is where you are in that shallow sleep. You're transitioning from being awake to being in a deep sleep. This is the beginning of your non-REM sleep. Delta is at the end of non-REM where you are in very deep sleep. Gamma is something that is not discussed in Brain Facts, but I wanted to add it in because it is one of the brain waves, and it is typically found in lots of sensory processing. If you are looking at a lot of information at once, there will definitely be some gamma waves recorded. Okay, now another huge topic in Brain Bee is neurons. So we're going to spend a good chunk of time breaking those down. So neurons are the cells of the nervous system. And what's unique about neurons with regards to other cells is the fact that they can transmit and they can receive electrical impulses or signals. So the way that they do that is because of their structure. The first structure to receive an impulse is called a dendrite. We also have the cell body or the soma. Within the cell body, we have the nucleus. So this is looking like biology, right? We have what we would call a somatic cell kind of in the middle, and then around that we have some unique structures that allow for electrical signals to be transferred. This long section here is called the axon, and around the axon we have little bubbles of myelin. Myelin is a substance used to insulate the axon to prevent electrical signals from dissipating and from weakening the overall transfer of those signals. That's how our signaling can be so fast, and that's how it doesn't take you long to pick something up, because that signal traveled quickly within fractions of a second. These little gaps between these bubbles of myelin are called nodes of Ranvier, and what they are useful for is excitatory conduction, which is the process of an electrical signal kind of jumping along the myelin so that it saves time instead of having to go through each bubble. And the cells that produce myelin are called Schwann cells. This is important to distinguish from oligodendrocytes. Schwann cells produce myelin in the peripheral nervous system, whereas oligodendrocytes produce myelin in the central nervous system. So an important distinction there. And then at the very end, we have the axon terminal, which releases what are called neurotransmitters. These are signaling molecules that will be transferred to the next neuron in what we call a synapse. So this is the general structure of a neuron, something that will definitely come up quite a bit in Brain B. There are two main types of neurons. We have excitatory neurons, which promote other neurons to activate. And moving forward, I'm going to call activating an action potential. An action potential is where the neuron gets excited and is able to generate its own impulse in order to elicit a response in another neuron. So 80% of neurons are excitatory, and the most common type is a pyramidal cell, and it has a pyramidal structure. So this diagram here is demonstrating an excitatory synapse, where we have glutamate, which is an excitatory neurotransmitter or signaling molecule, and it allows for the influx or the entering of positively charged ions into the receiving neuron. And that allows that neuron to activate and to get closer to an action potential. The other type of neuron is an inhibitory neuron that prevents other neurons from activating. 20% of neurons are inhibitory, and an example of an inhibitory neuron is a GABAergic neuron. So this is what an inhibitory synapse would look like. GABA stands for gamma aminobutyric acid, and it allows for a negatively charged ion to enter into the receiving neuron to prevent it from getting closer to an action potential. The important thing to remember about excitatory and inhibitory neurons is that they work in tandem. They work together to prevent overexcitation or overinhibition. So you cannot really have one without the other, even though there's a lot more excitatory than inhibitory neurons. An imbalance of those two types of neurons can lead to seizures and a condition called epilepsy. Okay, so another thing discussed in chapter 1 is glial cells. Glial cells are the helpers of neurons, and a lot of people get this confused, but glial cells are not less important than neurons because neurons cannot survive without glial cells. Yes, they have a more secondary role, but they are incredibly important. So there are 4 main types. One type is called an astrocyte. It looks like a star, hence the name astrocyte. It deals with synapse formation. A synapse is the process of a neuron sending its signal to another neuron. It provides the neuron with nutrients, and it regulates ion concentrations around the neuron to make sure that it's not too positive or too negative. We have ependymal cells, which produce cerebrospinal fluid and cushion the brain, the spinal cord, and peripheral nerves. And then we have the microglia, which are focusing on immune protection of neurons and overall the nervous system. And finally, we have oligodendrocytes, which I mentioned, that produce myelin, but only in the central nervous system. Remember that Schwann cells produce myelin in the peripheral nervous system. So these are the four main glial cells, at least in the central nervous system, that you would need to have a solid understanding of for brain B. Now let's talk about a synapse, which I've mentioned quite a bit. So when a synapse occurs, we get a transfer of neurotransmitters, which are signaling molecules from one neuron to another. And when that happens, we can have a change in membrane voltage potential, or just how positive or how negative the neuron is in terms of charge. So initially, before any activation, we are at resting membrane potential, which is typically negative 70 millivolts. Now, if a neuron receives neurotransmitters that are activating that neuron, we can end up with reaching an excitation threshold, which is at typically negative 55 millivolts. If we reach an excitation threshold, then there is no choice but for the neuron to depolarize. It's important that you understand that action potentials are an all or nothing process. They cannot happen unless you've reached the excitation potential. And if you have, the neuron has no choice but to activate. It's not like a maybe situation, it's a will, it's a must. So depolarization, if you think of polarization as being negative, then if we're depolarizing, that means we're becoming positive. So it's going from negative 55 millivolts to as high as positive 55 millivolts. Then we reach the peak, and on the way back down, once we have reached that action potential, we have something called repolarization. And repolarization occurs when the neuron is returning to that negative state. So it is coming back down in terms of membrane voltage potential. And usually what happens is, as it repolarizes, due to a delay in the closing of some ion channels, we end up with hyperpolarization, where it dips a little bit further below resting membrane potential, but over time it works its way back up to that negative 70 millivolts or so. So this is what an action potential of a synapse looks like. And this is super important for brain B as well. So let's talk about neurotransmitters. This is what a synapse looks like. This is what I mean when I say that one neuron is sending a signal to another. We have the axon terminal of the presynaptic neuron releasing neurotransmitters, which are signaling molecules, to the postsynaptic neuron, or the receiving neuron, which has receptors for those neurotransmitters. So zooming in, let's look at neurotransmitters specifically, which are, again, signaling molecules that can cause the postsynaptic, or the receiving neuron, to fire an action potential, or to get further away from an action potential. So it can either excite or inhibit the receiving neuron. Examples of neurotransmitters include glutamate, epinephrine, GABA, acetylcholine, and so on. They can also be reabsorbed by neighboring neurons to make sure that they don't continue to excite or inhibit after their work has been finished, and that is called reuptake. So there are two main categories of neurotransmitters. We have excitatory neurotransmitters, which are depolarizing the postsynaptic, or receiving neuron. So glutamate is excitatory. It's the main excitatory neurotransmitter. So it's the most common one that can be found in the nervous system, and it helps to make the receiving neuron more positive and potentially reach that excitation threshold and perform an action potential. We also have an inhibitory neurotransmitter, which repolarizes the neuron, so it makes it more negative. And the most common type of inhibitory neurotransmitter is GABA, or gamma aminobutyric acid. We have certain receptors for these neurotransmitters. So receptors receive these neurotransmitters and elicit a response in the neuron. There are 3 types of signaling molecules. There are hormones, for example, testosterone, oxytocin. Those are all hormones that can elicit a response in a neuron or a cell. We have neuromodulators, which suppress neurotransmitter release. An example of that is endocannabinoids, and we will talk about this in more depth in future videos. And we have prostaglandins, which do 3 things. One is to increase inflammation, increase pain tolerance, increase pain sensitivity, and potentially cause a fever. So it's not the most pleasant, but it's super important physiologically to protect you from continuing to injure a wounded body part because it hurts. Or it allows the body to fight against an infection. So there are 2 types of receptors. There are ionotropic receptors, which are typically faster, simpler, and just function with ion channels. As you can see in this diagram here, an ion channel simply allows for ions to enter the neuron to either make it more positive or more negative. And then we have metabotropic receptors, which are kind of the opposite. They're a lot slower, a lot more complex, and they can cause a variety of outcomes within the neuron. So if you want a more complex reaction in a neuron, you would typically activate a metabotropic receptor, and it can elicit an intracellular response. So it's also called a G protein couple receptor, and it causes a second messenger intracellular cascade to occur, and a more complex kind of reaction. So that brings me to the end of a summary of Chapter 1 of the Brain Facts textbook. Hopefully that helped to clarify a couple things. If you have any questions, please share them in the comments section. I'm always happy to help. And in our future video, we will be looking at Chapter 2 of Brain Facts, which is about senses and perception. Thank you so much for your time, and continue studying. I'll see you soon. Bye!